Canada. Xiaohong, thank you. And we have Dennis Chiraud, who is from Improve in France. And he has a very, very interesting research center there, which I hope to visit someday.
You're welcome.
So, thank you. So these are our judges. So we have some very strong judges here today. So now I would like to introduce our session progression, and that is that we have 6 students today. Each will have a few minutes to present the results of their research. They have an electronic poster, which you can view on the AOCS annual meeting website. So these students will talk for a few minutes, and then they will be given a chance to answer questions. We have a Google spreadsheet in association with this meeting that you can post your questions to, or you can send them to AOCS staff through a mechanism which is a mystery to me, unfortunately. However, there will be questions after each speaker, and we have a voting system which will allow us to vote. We'll have three presentations, and then there will be a vote. So each of the judges will fill in their matrix, and then the people who are watching, that's you, can also vote for your the person you chose did the best job. And then we'll go to the next section, which is the last 3 speakers, and then there'll be another vote session, and then the results will be tallied by our scorekeeper, and then we will announce the winner. We'll have a first place winner and a second place winner. Each of the participants will, of course, receive a certificate and will be noted on our website as participating in this competition, but the first and second places both have cash prizes, so that's kind of nice too. Okay, I will now introduce our first speaker. This is Mohammed Zaber at the University of Alberta, Canada, who has a presentation called Development of Chicken Feathers Derived Novel Adsorbent for the Removal of Heavy Metals from Contaminated Water.
Hello everyone. I want to start my presentation with the famous quotation of Benjamin Franklin. Once he said, "When the well is dry, then we will know the worth of water." He was absolutely right. According to World Health Organization, 2.1 billion people don't have access to clean drinking water, and 1.2 million people die every year due to unsafe drinking water. Although we are living in an industrialized world, but the amount of wastewater produced from industrial and agriculture sector is about 1,500 cubic kilometers, which is 6 times more water than available in all rivers of our planet. If we just talk about heavy metal contamination alone, arsenic has nearly affected 170 million people in 70 countries of our globe. Also, the existing technologies are either expensive, environmentally harmful, and have low absorption capacity. So we need a solution that should be cheaper, environmentally friendly, and can be implemented on a larger scale. Here comes my research project to decontaminate the heavy metal containing water using chicken feathers. Now you may be curious to know why specifically the chicken feathers. Chicken feathers are the poultry byproduct. Every year, hundreds of millions of tons of chicken feathers are produced around the globe, which are either landfilled or burned and creates environmental pollution. However, we can use them for productive purposes such as development of absorbent for water purification. Now comes the question how we can develop this absorbent using chicken feathers. The process is simple and pretty straightforward, and to answer this question, we need to understand the basic chemistry of the chicken feathers. Chicken feathers have mainly keratin proteins, which consist of disulfide linkage. By breaking these crosslinks, we can increase the surface affinity of the chicken feathers by introducing nanoparticles such as graphene oxide, so that they can easily capture or essentially stick to a variety of toxic contaminants, including heavy metals. To develop this adsorbent, we took chicken feathers, washed, oven dried, and then ground. Then these ground chicken feathers were dissolved using reducing agents. These dissolved chicken feathers were further chemically treated with the water dispersed graphene oxide to obtain the keratin graphene oxide mixture. This mixture was further subjected to dialysis and drying to get the adsorbent. After the successful synthesis of the adsorbent, we characterize this adsorbent using different techniques such as Fourier transfer infrared spectroscopy. This graph shows that the OS touching region, as well as amide 1, amide 2, and amide 3 region of the adsorbent, the intensity has been increased, and shows the presence of physical interaction between keratin protein of the chicken feathers and graphene oxide. This claim was further corroborated using XRD analysis. XRD analysis was basically used to see the crystallinity changes in the pattern of the chicken feathers after incorporation of graphene oxide. So, after introducing graphene oxide into the chicken feathers, the peaks are either reduced or disappeared. Also, the new crystallinity peaks are appeared that shows the presence of physical interaction between chicken feathers and the graphene oxide. Furthermore, scanning electron microscopy was performed to see the morphological changes in the chicken feathers after introducing graphene oxide. Chicken feathers have long shafts and smooth surface. However, after introducing graphene oxide, its surface turned into rough and shiny patches were observed due to the presence of the graphene oxide. Furthermore, these adsorbents were tested by using synthetic wastewater prepared in the lab with the help of ICP-MS. I'm pleased to say that we already did experiment for a proof of concept and currently working on the development of technology at a larger scale. And our study has 3 outcomes. Firstly, these heavy metals can be removed from 80 to 99% by using this adsorbent. And our adsorbent have maximum absorption capacity towards arsenic and selenium. Secondly, 1 kg of the adsorbent can remove the heavy metals in the 10,000 liters of wastewater. Lastly, and most importantly, our adsorbent have good adsorption capacity as compared to the existing technologies, as well as it is environmentally benign. At the end, I would like to say, with the development of this adsorbent, we can remove the potential health and environmental impacts even before their risk of occurrence. Thank you.
Thank you, Mohammed. That's a very interesting approach. I like your approach. There's a question. Actually, I have several questions, but here's the question that I will ask you. Do you think it's possible to recover the metals from the adsorbent? I would suspect. Please go ahead.
So, there is a possibility to recover the metals, and we are already planned to recover these metals. And if we recover these metals in a large quantity, of course, then we can sell these metals to the metal industries.
Thank you. Judges, do you have a question for Mohammed? Please speak up.
Yes. Do you want me to start, Scott?
Yes.
Yes,
that'd be great, Dennis.
I have 2 questions. Mohammed, thank you for the nice presentation. What's about food compatibility of graphene oxide Is it food grade material or not?
Well, there are some of the studies, they are saying it's safe, but however, some of the scientists, they said it's unsafe for the food. However, in our case, because our graphene oxide is the chemically linked with the adsorbent, so there are no possibility to transfer of the graphene oxide from the chicken feather into the water. So it's safe in case of water. We can use it for water purification purposes.
My second question was about what you explained. Don't we see leaks of this graphene oxide in the media? You know, after usage, cubic meters after cubic meters, can we leak and go through environment this product?
No, this graphene oxide is basically linked with the proteins. So I don't think so there is any possibility to the transfer of graphene oxide from the chicken feather into the water. And also we are using only very small quantity of graphene oxide. It's just 1% or 2 or 3%, not more than that.
Yeah.
Okay. And my last question is about the price. Do you have an idea, rough estimate of the treatment costs for, I don't know, 1 cubic meter, 1 gallon of water or whatever? Do you have an idea of these costs?
So yet we haven't do the cost analysis, but we are expecting that because our raw material is the waste. So we don't have any like the cost as such for the raw material. So we are hoping that that would be cheaper than the existing technologies.
Thank you, Denise. Do our other judges have any questions?
Yes, thank you for the great presentation. I have 2 questions. The first question is, may I know what parameters affect on the removal efficiency of the metals?
Okay, well, we actually worked on 2 parameters. The first one is the pH and the temperature. So there are other factors such as adsorbent amount are also play important role.
Okay, so at what pH, what is the optimum pH or temperature to get the maximum removal efficiency?
So we determine between 6 to 7 pH because we are also planning to use that one for the drinking water purposes too. So what we are doing is, if we are taking like the wastewater, we change its pH. We can increase or decrease. So we are using like between 6 to 7 pH. Whatever the result I showed, it's between 6 to 7 pH.
Okay, thank you. So my last question is, what's your opinion on the major challenge for scaling up this technology?
Because I'm using the bio-based adsorbent, so the existing technologies are mostly used the petroleum-based. So I would say I need to, when I'm going for a larger scale, so I need to consider that because it's a bio-based, while others are mostly petroleum-based products they are using. So that's the technology barrier to develop this technology at industrial scale.
Thank you.
Professor Wu, do you have any questions?
No, I just have a small question. I think, Mohammed, you did an excellent job in presenting your research. I really enjoy your presentation. So my question to you is, you mentioned about the cost of a feather is very cheap. I wonder about the cost of extracting the keratin from a feather and how that might, affect the overall cost of your technology. Thank you.
Yeah, so we haven't done yet the cost analysis, but for the chicken feathers, because chicken feathers are mainly consist of keratin proteins, up to 94%. So we are just extracting lipids from that, and the rest of material we are using. for the
Thank you.
absorbent.
Well, thank you very much, Mohammed, and thank you to our judges. We'll now transition to our next presenter. Our next presenter is Kaz Machida from the UC Davis in the USA. That would be California. His title is Effects of Enzymatic Extraction of Oil and Protein from Chickpea Flour on Protein Functionality. Kaz, would you begin?
Yeah, thank you so much. So from editing and revisions, I've realized my title is kind of confusing. So my new and improved title is Effects of Enzymatic Hydrolysis on Extractability of Oil and Protein and Protein Functionality from Full-Fat Chickpea Flour. So to give some background, we've seen an explosion in plant-based food options, including Impossible Foods, Beyond Meat, Just Egg. And to supply this increasing demand, chickpeas can be used for functional and digestible plant-based proteins. And to extract the chickpeas, the aqueous and enzyme-assisted extraction process can be used to get the proteins as well as the lipids. So the scope of this project was to fill a gap in the research. There's limited understanding of the effects of extraction conditions on extractability and the connection between that and functionality of chickpea macronutrients. So the goal of this study was to elucidate extraction parameters on the extractability and functionality. So to explain the process, we take full-fat chickpea flour that doesn't need to be defatted by hexane, and we mix it with water at a solid to liquid ratio of 1:10. We do the extraction at a temperature of 50 degrees, pH 9, and it's vigorously mixed for 60 minutes. There's 4 treatments that we use: the AAP, which is no enzyme, and the EAP, which uses protease at 0.5% for the 60 minutes, with EAP 2 and 3 undergoing 30-minute carbohydrates pre-treatment at pH 6. So the extracted slurries are then centrifuged and separated into the solids, which are the insolubles, and then liquid fraction, which is further separated into the skim and the cream and free oil. The chickpea we used was a steamed chickpea flour of the Kabuli variety, and our approximate analysis found that it had around 7% oil and close to 26% protein to begin with. So after conducting extractions on chickpea flour, we see that enzymatic treatments increase the protein and oil yields significantly compared to that of the AAP. Within the enzymatic treatments, we don't see a difference in the oil, and we only see a slight improvement in the protein yields. Additionally, molecular weight analysis showed that enzymatically treated proteins were significantly smaller in size. AAP proteins go up to 97.4 kilodaltons, while the EAP proteins only go up to 21.5. This was further confirmed by our degree of hydrolysis analysis, which showed that it showed an increase from 10.3 to 25.5%. This is most likely due to proteolysis of the proteins by the protease. And to test functionality, we first looked at solubility. At pH 4, we can see significant improvement with the enzymatic treatment. We chose pH 4 because chickpeas have a pI of 4.3, and this is important to us because this shows that enzymatically treated proteins can be applied in food applications of acidic conditions and not just basic conditions. Next, we looked at digestibility. That increased significantly as well, from 83.181 to 94.67% digestibility. We believe that the protease pre-digests the proteins and creates smaller peptides that are therefore more easier to digest and absorb. So in conclusion, we've been able to elucidate the impact of the AEP and the EAEP on chickpeas and seen an definite improvement in the yields for the EAEP. And we also saw functional analysis increase significantly, especially for solubility at acidic pH and digestibility of the EAEP proteins. And identifications of these extraction conditions is key to improving process feasibility and producing protein fractions for targeted food applications in the food industry. To wrap this up, I'd like to thank the USDA ARS Pulse Initiative for funding this project. I'd like to thank my PI, Dr. Juliana Bell, and Dr. Fernanda Diaz for their support, and to all my lab mates for their encouragement. And finally, thank you to the audience for listening to my presentation. Thank you so much.
Thank you, Kaz. A very interesting approach. I have so many questions, but I know that our judges are very curious as well. So I will limit myself to one, and that is, tell me which protease you used.
We use a Protex 6L, which is an alkaline endoprotease.
Okay. Judges, would you like to start?
I can start first. Tess, I enjoy your presentation. It's really interesting. My first question: why did you choose chickpea as your material for the research?
Well, our grant was given to us to study chickpeas, and also I believe that chickpeas are a good substrate for plant-based proteins because they have a mild taste. A lot of the legumes that are used sometimes have a very beany, very distinct taste, and that can come out in food applications where you don't want any kind of beany flavor.
All right. Maybe I missed some information. So when you extract the protein from your aqueous process, what's the protein content in your protein sample?
So the yields that I showed were
No, I was asking for the protein content, not the yield.
Of a specific fraction.
Yeah, I assume you get your cream fraction. You might also have your protein. What are you called? The sample you used for measure the protein solubility. So what's the protein content in the sample you used for measuring protein solubility?
So for protein solubility, we used the Dumas method. We take some freeze-dried protein and then we dissolve it in a solution and try to see a difference between the total protein and then the protein in the supernatant. And then the protein that we have in the skim especially was around 2%.
All right, thank you. I have a lot more questions, but I stop here now.
So maybe I can start asking. So thank you, Kaz, for this very interesting presentation. Do you know that most of the time your title is linking protein with functional properties, and most of the time functional properties are not only solubility or digestibility, but it can be also foaming, gelling, emulsifying properties. Do you have an idea of what is the impact of your process on these functional properties?
I believe that the foaming, from the literature that we saw, foaming properties, water absorption, oil absorption improve with enzymatic treatment, and I hope that I could do this, and possibly in the future. I think it should be done.
Thank you. adding enzyme in your extraction process will have a cost impact. Do you have any idea of the evaluation of this on your global process?
Yeah, so my next paper and hopefully my next presentation is doing economic analysis. And we actually saw that the enzyme in terms of large scale, it's very minimal in comparison to other factors like the cost of the flour, especially. And the enzyme we use, we from our cost analysis, we showed that like it's very minimal in comparison to the chickpea flour cost.
And my last question is about, again, like Jianping, the purity. You know, there are two products right now on the market at the industrial scale. And one is obtained by full fat extraction, and they reach 70% protein purity. And the other one is obtained after defatting of the flour, and they reach 85% protein purity. What is your level of protein purity in your extraction?
So we did not actually do any protein recovery, like any ultrafiltration or isolysis precipitation. So I cannot give you a specific protein purity. Hopefully that'll be a future study.
Okay, thank you.
Okay, thank you for the great presentation, Kaz. I really enjoyed it. I only have one question. So could you share some opinion on the limitation of this extraction method?
So the limitations of this extraction method is definitely the oil infiltrating the skim, which has the majority of the protein. When you have something with little oil like chickpeas, it's very hard to create a separation between the cream and the free oil and then the skim. So you get a lot of the oil that goes into the skim, and you can actually see that in the yields. The oil yield in the skim is considerably higher than the previous studies using the same process. And the other substrates that were used were peanuts and soy, which are much greater in oil content than chickpeas. And so because of that, like there's definite limitations. And I think a good step would be to try to improve upon the separation process and try to separate the lipids out of the proteins better. Okay. Because we that affects this the functionality as well.
Okay, just have a follow-up question, because I noticed during the extraction, the temperature is 50 degrees and the extraction time is 1 hour. So do you think this condition will negatively affect on the oil quality due to oxidation?
I think that is definitely a possibility. I think that's another future study that should look into that. And yeah.
Thank you.
Thank you, judges. You know, Kaz, we have an old expression, and that is that the larger the island of knowledge, the longer the shoreline of ignorance. And as you see here, you know, you do something, you present it, and so many questions. You've got years worth of material to look into. That's one of the reasons research is much more exciting than people generally realize. So we have our curiosity. Thank you very much, Kaz. We will have our third speaker now, and let me remind you that the voting for these posters and poster presentations is taking place in groups of 3. So after Rio is finished with her talk, we will have a pause for you who are watching to vote, and our judges can tally their votes. So our next speaker is Ryo Ogawa from Nihon University in Japan, and she will be speaking on examination of an efficient extraction method for rice bran albumin and its suppressive effect on postprandial blood glucose elevation. Ryo, would you start?
Yeah. Hello, everybody. My name is Ryo Ogawa. I'm a graduate student of Nihon University, Japan. Today, I'm happy to explain my research work on examination of an efficient extraction method for rice bran albumin that is suppressive effect on postprandial blood glucose elevation. First, I would like to explain the introduction of this work. In recent years, the number of the diabetic patients has been increasing worldwide. For diabetes, prevention by postprandial blood glucose control and early treatment are important. We have already shown that rice endosperm albumin, REA, suppresses postprandial hyperglycemia in vivo. And the mechanism is by adsorbing glucose like dietary fiber. On the other hand, rice bran is mostly used for low value added products. However, its rice bran is rich in protein. If the rice bran albumin, RBA, also has a function to suppress the postprandial hyperglycemia like REA, this will increase the value of rice bran. However, the yield of RBA from rice bran is quite low. The reason for its pro-extraction efficacy will be probably because of the binding of protein to starch and fibers. Thus, the hydrolysis of starch and fibers may increase the yield of RBA. In addition, RBA has been shown to adsorb glucose like dietary fiber in vitro and to have digestion resistance. It is expected to have suppressive effect on the increase in blood glucose level after meals. Therefore, in this study, we examined the effective extraction methods of RBA by using saccharifying enzymes. Then, we evaluated the suppressive effect of RBA on blood glucose elevation in vivo. Next, I would like to explain the methods. First, in the extraction method using enzymes, starch and fiber were hydrolyzed using hemicellulase, alpha amylase, and glucoamylase to extract proteins. Next, the reducing sugar content and the protein content in the extract were measured. Subsequently, a novel glucose tolerance test was performed to measure postprandial blood glucose and plasma insulin levels in vivo. The slides show the reducing sugars in the supernatant after various enzyme treatment in green and the amount of protein in red. The amount of reducing sugar in the extract became the highest when three kinds of saccharifying enzymes, hemicellulase, alpha amylase, and glucoamylase were all used. However, like the results of reducing sugar amount, the amount of protein was the highest when two types of saccharifying enzymes, hemicellulase and alpha amylase, were used. And glucoamylase treatment did not affect the protein content in the extract. Here is the results of blood glucose and plasma insulin levels in the OZTT. The blood glucose level increased sharply after glucose intake and gradually decreased to the original level over time. Compared with the control group, RBA laid the peak blood glucose level and significantly suppressed the increase in blood glucose level at 15 minutes after its administration, similar to REA. However, there was no significant difference in glucose ASE among the groups. The plasma insulin level also increased sharply in the control group at 15 minutes after oral administration of glucose. However, RBA significantly suppressed the increase in the plasma insulin level similar to REA. In summary, enzymatic hydrolysis of starch-and dietary fiber is effective for efficient extraction of RBA, and RBA suppressed the increase in blood glucose and plasma insulin levels after 15 minutes after oral administration of glucose. In conclusion, RBA from rice bran is expected to be used as a high value added component. Thank you for your attention.
Thank you, Leo. That was very interesting presentation and a very good suppression of insulin and expansion of the release across time. So I'm sure our judges have questions, but I have a question which maybe strike you as a little odd. But you know, when in Japan, when people are making sake, they can't generate the alcohol because of all these starches in the rice. So what they do is they incubate it with koji mold. And I wondered, have you thought about trying koji as a way of generating these interesting materials that you need?
Thank you for your question. Your question about why we don't use koji, right?
Well, I was just wondering if you have thought about using koji because it's cheap. It's just a suggestion. It's okay.
In the future, I want to try use koji. Yes. Thank you.
Good. Judges, do you have some questions for Leo?
I do. Leo, thanks for your very interesting presentation. It's always good to see a lot more research progress using rice albumin. I wonder, is there any difference between the albumin from rice endosperm as well and from rice bran?
Thank you for your question. According to the results of nano-LC mass meth, the amino acid sequence of REA and RBA were quite similar. However, as shown in this study, there is a clear difference in the effect of REA and RBA. So I would like to further examine the factors that make the difference between REA and RBA.
Very good. Well, as Scott just commented, after feeding albumin, obviously the insulin was suppressed, and your blood glucose level were also suppressed as well. So my question to you: how suppressing of your insulin also affect the reduction of your postprandial blood glucose level as well?
It's reason why glucose levels increase, so insulin levels decrease too, I think.
All right, thank you.
Rio, thank you for your nice, very nice and very interesting presentation. I have two questions. One is, in fact, you express all your experiment based on the RBA, which is for me, based on what I understand, an extract. You extract protein from rice bran. What is the purity of this protein extract? Because the question I have is what you observe linked to the protein or to other material that you also extract during your process?
Oh, thank you for your question. In animal experiments, RBA's purities are almost 100% in this study.
Okay, so it's extracted and purified.
Yes.
Okay. Do you think that the boiling, because in your extraction process, there is a boiling step with 5 minutes, which is usually impacting the denaturation of the protein. Do you think that it's an important element in your process, or do you think that native albumin without boiling will have the same impact?
Thank you for your question. RDA and RBA is some stable and do not denature even during boiling at 100 Celsius degrees for 120 minutes.
Okay, thank you.
Thanks for the great presentation. And by the way, I like your poster. I think it's really well designed and easy to follow. Yeah, I just have a quick question about your protein separation. In the extract, there is protein and the reducing sugar. So may I know what method did you use to purify the protein and use for the animal study?
So could you please repeat the question more slowly, please?
My question is, in the extract, it contains not only protein, but also the reducing sugar. So how did you remove the sugar and get the protein fraction for the animal study?
It's a dialysis.
Oh, okay. Okay. Thank you.
Thank you.
Thank you, Rio. Well done. And thank you to our judges. Now we will take a little pause for the voting to close for the first 3 speakers, and then we'll reopen a new voting option. So judges, if you care to tally yours or whatever you're doing, each of the judges have a matrix that they're filling in, looking at different aspects of the presentations.
So hello everyone, thank you for assisting to this presentation. So today I will present my research project which is about the health beneficial bioactivities of Canadian faba bean. So as you know, currently there is a growing demand for high quality and more sustainable protein sources. And in order to fulfill this demand, faba bean is a very interesting alternative because it contains a very high protein content. Also, it's a non-GMO, gluten-free, and not a regulated allergen, which are all characteristics that can be helpful in the food industry. However, faba bean is still underutilized, so its health-promoting properties are not well understood and well studied if compared to other widely used legumes. So this is why we have selected to investigate these properties through this project. So this is an overview of the methodology that was used. So in this project, 3 novel Canadian varieties have been compared. And they have been compared to 2 control legumes, which were pea and soy, because they are widely used in the food industry. So those legumes have been processed, and then they have been digested in vitro using the standardized InfoGest protocol. And after digestion, the small and highly active molecules have been enriched by ultrafiltration. And of course, these molecules included bioactive peptide. And then in the digested permeate, 3 bioactive properties have been investigated. So the antioxidant, antihypertensive, and anti-diabetic activities. And those were selected based on the result of an in silico analysis. So I will discuss some of the main, most important results of my research. So first, if we talk about the antioxidant activity, as you can see, several assays have been used in combination in order to target various mechanisms of action of antioxidant. So both in vitro chemical assays have been used, as well as a cellular model to assess this property in a more physiological relevant model. So for the chemical assays, there were no significant difference among the Faba varieties, except for the ABTS assays, as you can see here. And overall, the antioxidant activity of Faba was either similar or better than peas, or similar or lower than soy. For the cellular model, the outcome is slightly different. So as you can see, for the Faba and snowbird variety, the antioxidant activity was superior to pea and soy. So this is a very interesting finding. And this could be explained by the fact that the peptides that are generated by Faba may be more resistant to intestinal brush border, or they can as well have a better bioavailability. So of course, these elements will have to be further investigated. For the antihypertensive activity, the Faba had the Faba and Malic variety had a better activity compared to pea, but a lower activity than soy. And for the anti-diabetic activity, all the legumes investigated had an interesting activity. However, there were no significant differences among samples. So to conclude my presentation, as you can see, Faba has a very interesting bioactive potential, especially for antioxidant and antihypertensive property, and also especially if compared to peas. So eventually, Faba could be valued as a health-promoting food ingredient, but of course, further evaluation of this potential in vivo will be required. So thank you for listening to my presentation, and I will be happy to answer some questions.
Thank you, Delphine. I neglected to introduce Delphine. This is Delphine Martineau-Côté at McGill University in Canada, and she has spoken to us about Faba bean flour. So there are some questions. I was very surprised about the differences in your cellular antioxidant activities. They were striking, especially based on the peptide size. Are there many more different types of faba beans that would be tested, or I mean, you tested 5. How many are?
I tested 3 faba bean varieties,
Okay.
and 1 soy and 1 pea varieties just to compare.
Okay, that's right. I got confused there. So what platform did you use for your in silico predictions?
I used the BioPep database. So I used the proteins sequence of globulins of faba bean to verify if they were a bioactive fragment among this protein when they were digested with trypsin, pepsin, and chymotrypsin.
Thank you. Would any of our judges like to pose a question?
Yeah, I maybe can start. Thank you, Delphine. Very nice presentation. Very interesting because it's opening a lot of questions, not only on faba bean, but also on other pulses. Which protein, which type of protein extraction did you apply before going to this digestibility? Because you are speaking about globulin. Is it the full protein package, or is it just one fraction?
The globulin was just for the in silico analysis, but for my research for now, I just use a flour because we wanted to have the whole matrix in order to be more representative on how the faba bean will be consumed. But eventually, of course, it would be interesting to compare with purified protein to see if the same activities can be obtained.
Okay, great. And do you know if the faba bean varieties you were using were low in vicin and convicin, you know, because in terms of digestibility, you have the favism, and you need to pay attention to this because it can interact maybe with your experiment.
Yeah, of course. So the 3 varieties that I've studied were different. So the Fabel variety is low in vicin and convicin. So this is an important element. The Snowbird variety was low in tannin, and the Malik variety is high in both.
Okay, thank you.
Okay, probably I can go next. Your poster looks really great. Congratulations, great job. I have one question about your experimental design. Because your sample, the 3K permit, is mixture. It not only contains bioactive peptides, also include like polyphenols, right? So my question is why you use the in silico approach to screen the bioactivity only based on the bioactive peptides?
Yeah, so my hypothesis is that most of the bioactivity that I've observed are associated to bioactive peptide, but of course I have a complex mixture. So polyphenol and also small oligosaccharide can have an effect too in this aspect. But for my further research, I want to purify further my permeate and by size exclusion, and also want to identify the peptide present by MS to confirm that those effects are really attributed to a bioactive peptide.
Great, thank you.
Thank you very much, Delphine, for your nice presentation. I think I have 10 questions. I don't know if I have time to ask all of them. But I like to ask you the first question. When you did the digestion, okay, I assume that you're using acidic digestion. Did you also add the amylase in your digestion?
Yes, I used the InfoGest protocol. So I used the standard method. So I've used also amylase because there was starch in my extract, in my flour.
Okay, that's very good. So after digestion, so you have this digester, right? So what is the the protein content in your digest?
In my digest, in my permeate, or?
Yeah, I mean, after you finish your digestion, so you have digester, right? So
yeah.
What was the protein content in the digest?
Yeah, I've measured the protein. Well, because my permeate was in liquid form, so I've measured protein with the BCA assays. If I remember well, it was around 5,000 microgram per ml of protein.
Okay, let me ask the last question. Do you have a plan to measure the bioavailability of the peptides?
Yeah, this is my next step in my project. So I want to see if those peptides can cross the intestinal barrier using Charcot-2 cells. So just to see if those peptides can really have an impact in vivo after that.
Thank you.
Thank you, Delphine, and thank you, judges. We'll now take a short break until the next presentation. Our next speaker will be Yanting Shen, and she is at Kansas State University and is this year's winner of the Hans Konitz Award, the AOCS award that we give out every year, so congratulations. She will be presenting her poster, which is Modulating Intra and Intermolecular Interactions of pea protein isolate to improve its functional properties. Yanting?
Yes. Hi, everyone. My name is Yanting Shen. Today, I'm going to share my research about the modulating inter and intermolecular interactions of pea protein for its functional, improve its functional properties. Protein denaturation occurs along with the reactions to cleave the covalent bond, peptide bond, or break down the hydrogen bond or hydrophobic interactions. The sodium sulfate is a reducing agent that can break down the protein disulfide cross-linking. It will result the free SH groups. The urea denatures protein through the dehydrating protein molecules, and it will cause the repulsion between the protein by hydrophobic interactions and competitive hydrogen bonding. But the urea breaks down the hydrogen bond more efficiently than affecting the hydrophobic interactions. The SDS dominantly break down the hydrophobic interactions, and protease break down the peptide bond and result in the smaller peptides. So for this project, the objective of this study was to investigate the functional properties of pea protein isolate by modulating protein covalent and non-covalent interactions and understand the physical-chemical characteristics of the modified pea protein that are responsible for the functional change. So let's see our results. For the free amino group, we can confirm that the trypsin hydrolyzed pea protein had the highest free amino content. And for the free SH groups, with 1% of the sodium sulfate, the free SH content significantly increased compared to the control and other modifications. And for the surface hydrophobicity, 0.5% of the SDS modified pea protein exhibit the highest surface hydrophobicity among the others. The hydrophobic tail of SDS interact with the hydrophobic cones of the protein in the water, so it will result the largely exposure of hydrophobic side chains. For the SDS page under the non-reducing conditions, it is obviously to see that the protein modified by the sodium sulfate had overall lower molecular weight compared with others. And let's see the DSD result. The 1 mole of the urea modified pea protein had the highest denaturation temperature than the others. So that implies that the hydrogen bonding plays a dominant effect on the protein thermal stability. So next, let's look at our functional properties result. For the urea and SDS modified proteins have significantly higher values on the water holding capacities as well as on the emulsifying capacity and the stabilities. For the sodium sulfate modified protein have no significant improvement compared with pea protein. The trypsin hydrolyzed protein have significantly lower values on the water holding capacities, but it has greatly improved the solubility at the pH at the isoelectric point as well as good forming capacities. But for the oil holding capacity, all of the modifications have improved the oil holding capacities. And for the least gelation capacities, except for the sodium sulfate, the other modification methods are going to get a similar or even better gelation capacities than compared with control pea proteins. So last, we did the principal component analysis to further determine the relationship between functional, physical, chemical properties with different modifications. So we found that the improving the water oil holding capacities, emulsifying properties, and solubility at pH 7 are associated with the non-covalent interactions. The forming capacities and the solubility at isoelectric point improvement are associated with the enzymatic hydrolysis, which is attributed to the smaller peptides formed through the hydrolysis and greatly unfolded the protein peptide chains. In addition, breakdown the covalent cross-linking is associated with poor gelation capacities that is due to these associated the globulin protein and inhibit the disulfide exchanges. So in conclusion, this study provided the fundamental knowledge of the protein functionalities related to the protein covalent and non-covalent interactions, and it will contribute to the broader food applications of pea proteins. And that's all. Thank you for your listening. I'm happy to answer any questions.
Thank you, Yanting. A very good presentation. You've obviously done a lot of work. So
thank you.
I was wondering about your pea protein isolate, and I will be revealing my ignorance here. Do you expect trypsin inhibitor activity to be present in pea protein isolate?
Your question is trypsin will inhibit the activity. What kind of activity?
No, I'm sorry. Let me be more clear. Many of these pulses and grains and oil seeds have a particular protein in them which inhibits trypsin. So that has to be measured with a trypsin inhibitor assay. And I wonder about pea protein isolate. Do you know if that would be expected to have trypsin inhibitor activity in it? So that when you added the trypsin, it would have to titrate the inhibitor and then start working.
Oh, for this part, we haven't think about that, but maybe for our next step, we can do more research on this one for the nutritional aspect. But we did digestion for these modified proteins.
Okay, thank you. Judges, do you have some questions for Yanting?
Yeah, probably I can be first this time. Thanks for the great presentation, and I like the idea behind this project because there's already a lot of study work on understanding the relation between secondary structure and functionality. I think your work provides new insights on this. My first question is, because the hydrogen bonding stabilize the secondary structure of protein, and especially the alpha helix and the beta sheet, right? So my question is, why after using the urine to break down the hydrogen bond, but compared to the control protein, the secondary structure, including the alpha helix and the beta sheet, increased after this modification?
I think first maybe because of the concentration of the urea is not very high for this study, it's still low, and they have also competitive the hydrophobic interactions for the urea modifications.
Okay, so my next question is, do you think this denaturation is reversible or not? Because after the modification, you use the dialysis to remove the chemicals. Do you think that the interaction will reform?
Yeah, I think it may be reversible, but it will not be reversible after the heating process.
Okay, thank you.
Hi, Yanting. Thank you for this very nice presentation. I have two questions. The first one is about the protein hydrolysis you made. Do you know how far did you go for the hydrolysis in terms of percentage of hydrolysis? How many bonds did you break during this treatment?
The bond breaks still.
Yeah, when you make the protease action, you cut bonds between the protein.
Peptide bonds.
Do you know how many percent of the total bonds did you break? Was it a very deep hydrolysis or very slight hydrolysis?
Oh, I think it's a deep hydrolysis. It can be proved by our analysis from the free amino group. So if you compare with the control, so it have the I remember it's a 54 the m moles per grams of protein will be released for the free amino group. Compared with the control, it's only 11.
Okay, thank you. And the second question is, of course, you made a lot of lab experiment, but do you already imagine how you can translate this into a food processing application?
Yes, actually, this is only the fundamental experiment, but we hope to further use it in the food application. For example, the disulfide, the cross-linking. So if we use sodium sulfate to break down the cross-linking, we can use other food degrade reducing agent to treat it in the food and make it more different structures. For example, in the bakery industries, use like the cysteine to treat for the cookies and make more spread ratios.
Thank you.
Thank you, Yanting, for a very interesting presentation. I think your research is pretty fundamental, as you mentioned. So I'm going to ask you a very fundamental or basic question. Can you tell me why urea can break up hydrogen bonding in protein?
Why the urea break down the hydrogen bonding?
In protein, yes. Because that's the approach you used to damage the hydrogen bonding in protein, right? So why you're using the urea can disrupt or damage the hydrogen bonding in the protein structure?
Because in the previous studies, in the chemical studies, they all use the urea to break down the hydrogen bonding.
Oh, okay. So you refer to the literature. Okay, thank you. Thank you.
So yes, it's very important to connect your results and your research now with what's gone before. So that's a very common. Are there any further questions? We have one question from the audience, which is similar to one that Denise asked, which was, do you have thoughts on the practical application with all the chemicals for food grade pea protein. Let me start over. Any thoughts on practical application with all the chemicals for food grade pea protein processing? I would reinterpret this question as saying, can you use SDS in food? Can you use urea in food?
No, no.
Okay, so these are fundamental studies about the effects. Yeah.
Yes, yes.
But trypsin you could use, right?
Yeah, trypsin we can use.
Okay. Well, thank you very much, Yanting. I
Thank
think
you.
we are finished with our questions, and you can now turn off your camera, and we will go to our next presenter. We have one more student who will be presenting her work. And at the close of this, we will close our voting. So those of you who are watching, if you'd like to cast your votes, by the time Chidang is done, that would be great. So our next speaker is Chidang, and Chidang is at the University of Ottawa in Canada. And the title of her poster presentation is Bioinformatics Analysis of Adhesin-binding Potential and ADME Tox Profile of Anti-Helicobacter pylori peptides derived from wheat germ proteins. Chi, please go ahead.
Welcome everybody. My name is Chi Dang and I'm an undergraduate student at the University of Ottawa. And today I would like to present my research project titled Bioinformatics Analysis of Adhesin-binding Potential and the ADME Tox Profile of Anti-H. pylori peptides derived from wheat germ proteins. Okay, so firstly, H. pylori is an important human pathogen causing gastric infection and possible ulcers of over 50% of the world's population and has been classified as class 1 carcinogen by the World Health Organization. And the infection is usually treated with antibiotic regimen. However, due to the emergence of multi-drug resistant bacteria, it is important to explore alternative treatments, and anti-adhesive therapy with bioactive peptides is considered one of the most promising strategies by interfering with the bacterial adhesion to the host cells. And food-derived bioactive peptides are gaining more interest as nutraceutical candidates, as they have been demonstrated to have therapeutic potentials. And there are also some research that prove that they are optimistic anti-adhesive agents. And so the objectives of my study were to validate in silico the anti-adhesive property of the previously identified wheat germ proteins, and then to evaluate the ADME tox profile based on the pharmacokinetics parameters. So starting with the initial database of 267 peptide sequences, we perform in silico analysis with digestion with pepsin, which is stomach enzyme at physiological pH to assess the biostability. And then we perform molecular docking to visualize the binding as well as to obtain the binding affinity of the peptides to the two most dominant H. pylori adhesins, BAP-A and SAP-A. And then we evaluate the predicted pharmacokinetics, physiochemical properties, as well as toxicity of the final 16 selected peptides. So the results here of the representative 3 peptides extract from the wheat germ proteins. And so you can see that there are multiple binding poses of the peptides to the adhesin SAP-A, as well as so they bind with medium to strong binding affinity. And the peptides were shown to be a poor oral bioavailability due to the high number of rotator bond as the and the polar surface area. And so this could inspire more further research. As well, they have high solubility, and all are shown to be non-toxic. And they are also have low gastrointestinal absorption, which is desirable in this context, considering the targeted application of the peptides were to act as receptor analogs to prevent H. pylori infection. And so the bioavailability radar image shown over here of the peptides are quite similar to each other, and they're also similar in many parameters to the 3-sialylactose sodium salt, which is a compound derived from human and bovine milk that has been verified binding capability to SAP-A, as and also being involved in some clinical research in human and rhesus monkey. And so this suggests that the peptides could possess the same mechanism of action as well as biofunction in preventing H. pylori infection. And so in conclusion, the anti-adhesive property of the defatted wheat germ-derived bioactive peptides was validated. And based on the in silico ADME tox profiles, it is shown that they show the promising anti-adhesive activity. And this is my reference, and thank you for listening. I will be happy to take any questions.
Thank you, Chi. Very interesting talk and a very fun approach to this. So I have a question about your docking. So you carried out in silico digestion and you derived some peptides. I would expect that the digestion would take place in the stomach, right? Because it's a pepsin is a stomach enzyme. But suppose there could be some carryover into the intestine. So when you did your visualization of your docking, did that happen at stomach pH or small intestine pH, or would you expect it to not make any difference?
We perform docking with our regards of the pH. We only perform like the assessment of biostability with the pepsin at physiological pH. So that was the previous step to docking. And so that could be what we look into further, but what I predict that the low pH of the stomach, if it doesn't affect the peptide stability, but in the intestine, I think that the pH is a bit higher. So it could alternate the structure of the peptide. So we could also look further into that too.
Thank you. Do our judges have questions?
Yes, maybe I can start. So thank you, Chief, for this very nice presentation. Very, very interesting. When you speak about peptides, do you know the molecular weight of these peptides you were using?
Yes, so our molecular weight, just give me 1 minute, please. So our molecular weight are pretty high, so they're all higher than 500. So it kind of violates the Lipinski rule of 5. However, so they range in about maybe 800 to 1200. So it's pretty big, yeah.
Yeah. And my second question is about the purity of these peptides. Are you using really highly purified peptides, or it's just an extract containing maybe some side molecules?
Yeah, so we extracted from, so we have like a hydrolysate from the wheat germs, and then we perform SDSPH as well, blotting, and then identify the peptides components by mass spectrometry. So of course that there could be some impurity that involved, and so this could also affect the properties that we predict in silico. And so from the next step, we could validate in vitro, and then we could also look into the purity of the samples that we collected before too.
Okay, thank you.
Okay, I can go next. So Chida, I think you did an excellent job. I wanted to know that you are actually an undergraduate student. Great job. Okay. My first question to you, where did you get these 267 peptides?
Yes, so I did this based on the initial database. So there was a study before from one of the postdoc fellow in our lab. So she did, so it was like in vitro experiment, and then they do like identification of the peptides based on the experiment. And so for the in silico work, that was the database that I worked on. So these are already verified with some properties against H. pylori infection.
Very good. Could you tell me how long did you spend on this project to date as you presented?
Yes, so because it's bioinformatics work, so it's a lot faster than if you do them with laboratory. So in terms of many that I use in these are web-based tools. So maybe like Streamline takes like a couple of weeks to like a few months. And then the docking part is the longest part, but in general, it is very fast to obtain result.
Okay, thank you. I know that's also need a lot of intelligence as well. Could I ask you what ADME stands for?
Yes, so ADME is the pharmacokinetics parameters of absorption, metabolism, distribution, metabolism, and excretion.
Excellent, thank you.
Thank you.
Hello, Chi. Yeah, I agree with Dr. Wu's comments. As an undergraduate student, I think you did a wonderful job. Yeah, well done. I just have a quick question. Could you briefly explain when you do the in silico digestion analysis, why you only use the pepsin and didn't choose the enzyme from the intestine?
Yes, so the reason why we use pepsin is that H. pylori infection usually, because of the flow of the stomach, it's usually concentrated in the bottom of the stomach. And so in that area, pepsin is according to what I know, is the only protease available in that area. And so if there are any activity that we want the peptides to be, we would assess with the pepsin only. Yeah.
Thank you.
Thank you.
Thank you very much, Chi. Thank you very much to our judges. We will be finished with our poster presentations, and we thank all of our students, and we will have a short pause where the judges will be able to enter their results and send them to our scorekeepers. So I would like to tell you about the sessions that are going to be carried out in the AOCS meeting for the rest of the meeting that have to do with proteins and co-products. So tonight, 7 p.m. Chicago time, there will be a talk called Updates on Protein Research in China, and that will be chaired by Keshin Liu, who was recently awarded the award of a fellow for his significant work in this field. He's truly a leader in the field. On Monday at 7 a.m. Chicago time, we're going to have a presentation session called Protein Biofunctions. And that is a session together with the Health and Nutrition Division. Also on Monday at 4 in the afternoon, Chicago time, we're going to have a session called General Protein and Co-Products. So that will have quite a variety of talks it can be very interesting to look at these different aspects. On Tuesday at 10 o'clock in the morning, we'll have some presentations. One of them is the Lifetime Achievement Award, and there'll be a lecture graduate student oral competition. So that'll be very interesting. That again is Tuesday at 10 a.m. On Wednesday next week at 6 a.m., there'll be a session called Application of Advanced, Novel, and Clean Tech Processing for the preparation and utilization of plant proteins for foods, Part 1. And that's a very long session, so that's going to be very, very broad and very practical. And Part 2 will be the next day at 6 o'clock in the morning. So applications in food, if you have an interest in that, this is a really good session for you. Finally, on Friday, we have 2 sessions. One, we're going to have a presentation from an association in Europe called Bridge to Food, which brings together a lot of high-level food company executives. And so Gerard Essink is going to be speaking on Research Investment Agenda, Plant-Based Foods and Proteins 2030, developing the roadmap. Gerard is a very dynamic and forward-looking individual, so this could be a very interesting talk. And then finally on Friday, next Friday, a week from today at 10 a.m. will be a session entitled Emerging Sources of Protein. So I encourage you to catch as many of these as you can, because one person talking will represent 3 or 4 months of research, and it's much faster than doing all the work yourself. Thank you very much for your patience. And we have a tie for second place today. And both Kaz and Yanting were scored the same. And so very strong presentations. And so you will each receive the second place award. And our first place winner is Delphine. So we thank you very much for your participation. And I will tell you what I like to say to everybody. We have a saying in the martial arts, you learn more from your failures than you do from your successes. So while none of you can consider this a failure because you all did very well, you can also study what the others did and do better next time. So thank you very much to the judges. You asked very insightful questions and we appreciate your time and attention because I know you're all very busy, and appreciate the way you honor the students by showing up here and interacting with them. Thank you to the students. We wish you long, successful careers, hopefully in connection with the American Oil Chemist Society. Thank you, everybody.
Thank you. Thank you, all the judges.